The next 5 chapters are going to be concerned with the science of neurobiology, or the study of the nervous system. This system is of great complexity and mystery. It's the foundation of all our conscious experience, our personality, and behavior. The nervous system has intrigued biologists, physicians, psychologists, and even philosophers for years. Understanding it seems to be the ultimate challenge facing the behavioral and life sciences. In this first chapter, we begin the study of the simplest organizational level, the nerve cells or neurons, as well as their supporting cells, the neuroglia. We'll examine some electrophysiology in neurons, as well as how synapses work, how the neurons communicate with each other, and then a little bit of insight into how we integrate these signals into experiences. If the body needs to maintain homeostasis and function effectively, all of its trillions of cells need to function together. It's the nervous system that allows us to do this. The endocrine and nervous system together maintain our internal homeostatic coordination. The endocrine system is a system that communicates by means of chemical messengers or hormones that are secreted into the blood and circulate throughout the body. The nervous system works much more quickly. It employs electrical and chemical means to send messages from cells to cell. The nervous system will carry out its task in 3 basic steps. The sense organs receive information about changes in the body and the external environment, and then transmits coded message to the spinal cord and the brain. The brain and spinal cord then process this information. It relates it to past experiences and determines what response is appropriate to the circumstances at hand. Then the brain and spinal cord issue commands to the muscles and gland cells in order to carry out the response that's necessary. The nervous system has 2 major anatomical subdivisions, the central nervous system and the peripheral nervous system. The central nervous system consists simply of the brain and spinal cord, and they are enclosed by bony coverings like the cranium and the vertebral column. The peripheral nervous system is everything outside of those bony coverings. It's composed of nerves as well as ganglia. Nerves are bundles of nerve fibers, the axons of many, many nerves, wrapped in fibrous connective tissue. A ganglion is a knot-like swelling in a nerve where the neuron cell bodies are concentrated. This chart shows us the basic breakdown. We have the central nervous system and the peripheral nervous system. The central nervous system consists of brain and spinal cord. Then the peripheral nervous system, we're going to look now at the sensory and motor divisions, each of which, as you can see, are subdivided further. The sensory or afferent division carries sensory signals from the various receptors in our periphery to our central nervous system. It informs the central nervous system of the stimuli within or around the body. The sensory division is divided into 2 parts. The somatic sensory division, which carries signals from receptors in the skin, muscles, bones, and joints, and the visceral sensory division, which carries signals from the viscera of the thoracic and abdominal cavities, things like the lungs, stomach, heart, urinary bladder, kidneys, so on and so forth. So the sensory division here, we've just seen the visceral and somatic sensory. Now let's go look at the motor division. It also has a visceral and somatic component. The motor or efferent division carries signals from the central nervous system to glands and muscles that carry out the body's responses. These target cells are called effectors. They're the cells and organs that respond to the commands from the central nervous system. The somatic motor division carries signals to the skeletal muscles and elicit an effect at the target. For example, it might produce muscular contraction. Some somatic reflexes are involuntary muscle contractions that are also under control of the somatic motor division. The visceral motor division, motor implies output, right? So the visceral motor division carries signals to the glands, cardiac muscle, and smooth muscle, and all the viscera of the abdominal, pelvic, and thoracic cavities. As a whole, we call this the autonomic nervous system. The autonomic nervous system is further subdivided into sympathetic and parasympathetic. The sympathetic division is the fight or flight division. It tends to arouse the body for action. It accelerates the heartbeat and respiration rate while inhibiting digestive and urinary systems, systems that we don't need to use during the fight or flight response. Get shut down. Systems we do get turned on by the sympathetic division. The parasympathetic division is the visceral motor division that is the rest and digest system. It tends to have a calming effect. It slows the heart rate and the breathing, whereas it stimulates the digestive and urinary systems. We don't need such great circulation in the muscles when we're resting and digesting, so blood is delivered to the digestive organs. Again, this is a great figure to review the divisions of the nervous system. First, central and peripheral, and then we have the sensory and motor division and the visceral and somatic divisions of each, as well as the sympathetic and parasympathetic divisions. Close your book quickly and see if you can diagram this out without even looking in your notes. Go and fill in the gaps. When you've got as much as you can down, look in your notes, fill in the gaps, and then do it again until you can get all of these divisions down packed. The communicative role of the nervous system is carried out by the nerve cells or neurons. These cells have three fundamental physiological properties that enable them to communicate with other cells. First is excitability. All cells are excitable, that is, they can respond to environmental changes called stimuli. Neurons have developed this property to the highest degree. Second property is conductivity. Neurons respond to stimuli by producing electrical signals that are quickly conducted to other cells at distant locations. And the third is secretion. When the electrical signal reaches the end of the nerve fiber, a chemical neurotransmitter is secreted and it crosses the gap and stimulates the next cell. There are three general classes of neurons determined by their functionality. There are sensory or afferent neurons. They're specialized to detect stimuli and then transmit information about the stimuli to the central nervous system here. They begin in almost every organ of the body and end at the central nervous system. Afferent means to conduct signals towards the central nervous system. The next group are interneurons or association neurons. You can see here, interneurons lie entirely within the central nervous system. They receive signals from many neurons and carry out an integrative function. They process, store, and retrieve information, as well as making decisions about that information and determining how the body will respond to the stimuli. 90% of all neurons are interneurons. They lie between and interconnect the incoming sensory pathways and the outgoing motor pathways of the central nervous system. Although it's pictured in a very simple way here, there could be many different interneurons involved in the processing of this signal. The third class are the motor or efferent neurons. They send signals out to the muscles and gland cells, or the effectors. They're motor because most of them lead to muscles and have an effect on movement. Efferent means conducting signals away from the central nervous system. Now let's take a look at the structure of the neuron. There are several different varieties of neurons, but for the purposes of looking at the anatomy of a neuron, we'll look at a spinal cord neuron. The soma is the control center of the neuron. It's the cell body. It's also called the neurosoma or the perikaryon. It has a single centrally located nucleus with a very large nucleolus. The cytoplasm contains the mitochondria, the lysosomes, a Golgi complex, numerous other inclusions, and an extensive rough endoplasmic reticulum, as well as cytoskeleton. The cytoskeleton consists of a dense mesh of microtubules and neurofibrils, which are bundles of actin filaments. These compartmentalize the rough ER into dark staining regions called Nissl bodies. Nissl bodies are unique to neurons and are a helpful clue to identifying them in tissue sections when they're mixed with other cell types. Mature neurons contain no centrioles, and thus they'll have no further cell division. Other major cytoplasmic inclusions in a neuron are glycogen granules, lipid droplets, melanin, lipofuscin, which is a golden brown pigment that's produced when lysozymes digest worn-out organelles. Lipofuscin accumulates with age and pushes the nucleus to one side of the cell. The granules are also called wear and tear granules because they're most abundant in old neurons. The soma usually gives rise to a few thick processes that branch into a vast number of dendrites. They're named for their striking resemblance to bare branches of the trees in the winter. The dendrites are the primary site for receiving signals from other neurons. The more dendrites a neuron has, the more information it can receive and incorporate into its decision making. These dendrites provide a precise pathway for the reception and processing of neural information. Finally, the axon, or the nerve fiber. It originates from a mound on the side of the soma called the axon hillock. The axon is cylindrical and it's relatively unbranched for most of its length. However, on occasion, we'll see axon collaterals branching off the main axon. Most axons branch fairly extensively at the distal end. For example, as the one single axon innervates many different nerve fibers, there need to be a number of different branches. The branching that we see at the terminal end is called terminal arborization, and each terminal has a synaptic knob or a terminal bouton, which is a little swelling that forms a junction or synapse with the next cell. The axon is specialized for rapid conduction of nerve signals to points remote from the soma. The axoplasm is the cytoplasm of the axon. The axolemma is the plasma membrane of the axon. And despite all the branching, there's technically only one axon per neuron, and it's wrapped in a myelin sheath that's created by Schwann cells. The myelin sheath helps it conduct signal along its long length. Each synaptic knob contains a number of synaptic vesicles that are full of the neurotransmitter that will be dumped into the synaptic cleft in order to signal the postsynaptic neuron. Take a moment here to diagram a simple neuron, including aspects of the cell body, the dendrites, and the axon. So not all neurons fit the preceding description. They're classified structurally according to the number of processes that extend from the soma. Multipolar neurons have one axon and multiple dendrites. They're the most common type of neurons, and they constitute most of the neurons in the brain and the spinal cord. Bipolar neurons have one axon and one dendrite. These are cells like the olfactory cells, the cells in the retina, as well as those in the inner ear. Unipolar neurons have a single process that leads away from the soma. A short distance away from the soma, the process branches like a T. In most other neurons, a dendrite carries signals towards the soma and the axon carries them away. In a unipolar neuron, there's one long fiber that bypasses the soma and carries signals directly to the spinal cord. These type of neurons in general will lead from the sensory receptors in the skin and the organs to the spinal cord. The final type are anaxonic neurons. Anaxonic neurons have multiple dendrites but no axon. They communicate through their dendrites and they do not produce action potentials. These can be found in the brain, retina, and adrenal medulla. So neurons are very large cells. These long axons need to have nutrients passed down through them. All the proteins that are needed are made in the soma, where you have all the protein-making machinery, such as the ribosomes and the endoplasmic reticulum. So how do we get the proteins and materials from the cell body down to the very end of the synaptic knob? Materials can move down the axon away from the soma, or they can move back up towards the soma. These are called anterograde transport and retrograde. Retro is to go back. Microtubules guide materials along the axon. They're actually little motor proteins called kinesin and dynein, and they carry materials on their backs as they crawl along the microtubules, literally propelling themselves like little motors along these road-like microtubules. Kinesin are the motor proteins in anterograde transport, and dynein are the motor proteins that are involved in retrograde transport. There are two types of axonal transport. There's fast axonal transport and slow axonal transport. Fast axonal transport occurs at a rate of about 20 to 400 millimeters a day, so it's not really that fast. 400 millimeters is about 40 centimeters or half a meter. It's not even the length of your leg. In fast anterograde transport, we'll see mitochondria and synaptic vessels and other organelles being moved, as well as calcium ions and enzymes like acetylcholinesterase, which is used in the breakdown of acetylcholine. In fast retrograde transport, we're going to see the return of used synaptic vesicles and other materials to the soma. These materials will inform the soma of the condition of the axon terminals. Some pathogens exploit this process to invade the nervous system. They enter the distal tips of the axon and travel to the soma by retrograde transport. Tetanus toxin and herpes simplex, rabies, and polioviruses use these mechanisms to get into the cell bodies of neurons. The delay between the infection and the symptom time is how long it takes for these invaders to be transported up to the axon. In slow axonal transport, which is also called axoplasmic flow, the rate is only about half a millimeter to 10 millimeters a day, so that really is slow. And it's always anterograde, meaning towards the axon terminals. It moves enzymes and cytoskeletal components down the axon. It renews worn-out axoplasmic components in mature neurons and supplies new axoplasm for developing and regenerating neurons. Damaged nerve fibers will only regenerate at a speed that's governed by the slow axonal flow rate. So again, fast transport can go either anterograde or retrograde, and slow can only go anterograde. That brings us to the end of our first section for this chapter. Next, we'll look at the supportive cells of the nervous system, the neuroglia.